Hello guys, welcome back to my channel. Sorry I did not post anything for I think last 1 month. I was pretty busy, so I did not make any new videos. But anyways, so if you remember, we were studying about the DNA replication, right, and all the processes. So from most probably today, so I will try to post each and every single day. That's one plan, right? And like as you know, like whenever we do a topic, so we finish the entire topic and then I post like maybe a 30-minute 40-minute video, but I'm planning to do in small parts because I find maybe that might be are if you are like studying continuously or watching a video that gives you a lecture, and I'm not sure, it might get boring. So today we'll study DNA repair. So what my plan is, so so this is a complicated diagram. So I already draw this to explain DNA repair. So there are different parts of DNA repair. So we'll get into it, and I will try to explain with it. So DNA repair. So when this comes into our mind, why we need DNA repair, right? So there is a DNA replication process, and the DNA replication process itself makes sure that the DNA that is replicated should not be damaged, or should not, the base pair should be corrected, right? So all the entire DNA replication process is proofreading and making sure that the DNA, new DNA strand that is being made, it should be without any mutations, right? So maintaining the genetic stability that an organism needs for its survival requires not only an extremely accurate mechanism for replicating DNA, but also mechanism for repairing the many accidental lesions that the DNA continually suffers, right? So that's why we need DNA repairs. So most such spontaneous changes in the DNA are temporary. So whenever there are like most sudden changes in this DNA strand, so they are temporarily because they are immediately corrected by a set of processes that are collectively called the DNA repair. So there are like whole bunch of protein mechanisms and enzymes that helps to maintain the DNA properly. Like whenever there is any damage in the DNA, that proteins and enzyme tries to repair it. So of the 10,000s of random changes created every day in the DNA of a human cell, like for example by heat, metabolic accidents, radiations, right? So DNA is damaged by heat, right? Metabolic, so there might be metabolic accidents. That's the reason that DNA can be damaged. There are like radiations, various sorts of radiations, ultraviolet radiation, even exposure to so there are like also exposure to substances, right, in the environment. So there are some kind of substances in the environment that can also damage your DNA, right? So there are like many mechanisms that can damage the DNA, right? And only a few. So of all this mechanism that is happening and trying to damage your DNA, of all this, so there are only few, maybe less than 0.2% accumulate as a permanent mutation in the DNA sequence. The rest are eliminated with remarkable efficiency by the DNA repair. Right? So, recent studies of the consequences of the diminished capacity for the DNA repair in humans have linked many human diseases with decreased repair. So, so you can see a table on your screen. So that shows that the defects in human gene whose product normally functions to repair the mismatched base pairs resulting from DNA replications error can lead to an inherited predisposition to cancers of the colon and some other organs, right? So you can see in the table, like there is a name, what type of it, there is a phenotype, and there is this enzyme or process that are affected, right? So like for example, there is in another human diseases, as you can see, xeroderma pigmentosum. The afflicted individual have an extreme sensitivity to ultraviolet radiation because they are unable to repair certain DNA photoproducts. So this repair defects results in an increased mutation rates that lead to serious skin lesions and an increased susceptibility to skin cancer, right? So there is this type of damage. Finally, there are like mutations in BRCA1 and BRCA2 genes compromise a type of DNA repair. So this compromises a type of DNA repair known as homologous recombination and are a cause of hereditary breast and ovarian cancer. So these are some of the reason, right? And these are some type of the DNA repairs, right, that are damage that causes some diseases. So just like any other molecule in the cell, DNA is also continually undergoing thermal collisions with other molecules. So that results in sometimes major chemical changes in DNA. So as we know, there is a cell, there are a whole bunch of different molecules in the cells, right? So as all other, DNA is also having that thermal collision with other molecules, and sometimes it results in the major changes in the DNA. So for example, DNA of human cell loses about 18,000 purine bases. So purine, if you don't remember, so that is adenine and guanine. All right, I will write it down. So it loses purine bases, and that is adenine and guanine. All right, so these are purine bases every day. So the human cell loses 18,000 of purine bases from the DNA strand every day. Why? Because the N-glycosyl linkage. So this is the N-glycosyl linkage to deoxyribose hydrolyze a spontaneous reaction called depurination. So there are two. First DNA, there is damages. So that is by hydrolysis. So this one, right? So this one, the first one is depurination. Why? So as you can see, so this part, just this part, if you're able to see this, so just this part is just a normal, for example, that is one of the bases of our DNA, right? So phosphate, sugar, and the base, right? So that is guanine, right? So there are like some hydrolysis happening as there are thermal collisions happening in those cells. So sometimes there is like 18,000 per day in cell. Some bases loses this adenine or the guanine due to this hydrolysis reaction. It removes this base, and the net result will be the DNA will be missing guanine. So there is no base and it is missing the base. So that's one. And the second is deamination. So the deamination is similar to the depurination. So this is a spontaneous reaction, and that happens around like hundreds bases per cell per day. So in this reaction, it's the same thing. There's a cytosine. So what happens in this? Due to hydrolysis, the cytosine is replaced by uracil. So that is replaced by uracil in this reaction, and like the DNA strand will have uracil. You know that the DNA should not have uracil, as uracil is with the RNA only. Right, so these are two of the damages that are happening every single day. So 18,000 per day that are purine bases are missing. For this, like 100 bases per cell per day. So DNA bases are occasionally damaged by an encounter with reactive metabolite produced in the cell, including reactive forms of oxygen and high energy methyl donor as well. So that is as adenosyl methionine, or by exposures to chemical in the environment. Right, so as I said, so due to the ultraviolet radiation from the sun, so that can damage thiamines. So the third one is thiamine. So this is the first one is a thiamine normal. So that's, for example, our DNA sequence, and both. So first and the second, for example, this have both thiamine beside each other. So what happens due to the ultraviolet radiation of the sun, that can produce a covalent linkage between these two adjacent pyrimidine base in the DNA that will form this thiamine dimer. So what will happen? So this both thiamine will be linked by covalent each other. So this is monomer. So as both are linked, so it will form thiamine dimer, right? And most of this changes, depurination, deamination, or thiamine, would be expected to lead either to the deletion of one or more base pair, or to a base pair substitution in the daughter DNA chain. So for example, if this is not repaired, right, so when the DNA is replicated, it will be missing a base pair, right? In this, when it is not repaired, so it will have a wrong base pair, that is uracil. It should not have that, and this will have a covalent linkage with each other, right? So the mutation would then be propagated throughout the subsequent cell generation. So once the mutation is there, so it will go along the generation, right? So such a high rate of random changes in the DNA sequence would have disastrous consequences. So these are few of the examples. So there is a list, like what can happen or what type of damages are happening to the DNA. So you can see the list that the DNA lesions. So we saw depurination, depyrimidation, cytosine deamination. We saw that, right? So there are oxidation as well. Non-enzymatic methylation happens. So these are all the DNA lesions, and it also shows number of repair in 24 hours, right? So this much has been repaired. So this, like for example, 18,000 are repaired every single day, right? So this is our first part of the DNA repair, right? So we just covered the what are DNA repairs and what type of DNA damages happens to the DNA. So these are some of the DNA damages that are happening to our DNA. In the next or the second part, we will see the DNA repairs mechanism. So different methods of DNA repair. All right, we'll see you in that.